This is an introduction to genetics. Oftentimes, it's referred to as Mendelian genetics, after the individual that kind of brought it to the surface back in the 1800s. That was Gregor Mendel, an Austrian monk. And what we'll like to do is look at what Mendel did and who he was. He's oftentimes known as the father of genetics. I don't know if this is a good term, but again, he was one of the ones that brought it up to the surface for people to review his work. And I would say that he looked at how traits were inherited. Specifically, he looked at pea plants. Pea plants were readily available at his time, and he was just a good, keen observer. He had some good math skills and recorded all of what we would say is he followed the scientific method. And as a result of these keen observations, he was able to make predictions. And that's what we look at today, as genetics is the study of genes, and this idea of heredity is the study of being passing these genes from one generation to the next. Oftentimes, we just refer to this as inheritance. You've inherited a certain gene, perhaps by your parent, and you will maybe pass that on to the next generation as well. So, before anybody knew anything about DNA, Gregor Mendel began to look at patterns of inheritance in pea plants. And he looked at a variety of characteristics, from shape to size to coloration of the pea pods. And his discoveries became really what we would refer to as the foundation of a field that we call genetics. So, what did he do in this time? Again, I have a time period down below there where it's 1822 to 1884. Again, some of his contributions. And I will tell you that some of his contributions got lost and then rediscovered in the late 19th century. So, for a period of about 50, 60, maybe years, Gregor Mendel's work was lost to science and then kind of rediscovered again and kind of brought our modern day approach to genetics with some of the work that he had contributed to. So, what he did was this. Let me give you an example. Mendel crossed pea plants, and he looked at specifically one trait at a time. And he crossed white flowered plants with purple flowered plants. And we would refer to these as the original or the parent. So this is something that is um, the P standing for the parent generation. What I'd like to look at is excuse me, what do you hypothesize the offspring or the next generation, which we call the F1 generation, what would it look like? In other words, you cross a purple and a white, what would the next generation be? And this next slide kind of shows you how he did it. And it's numbered 1 through 5. And if you follow along on the side here, you'll notice that um, plants oftentimes have both the male and female parts to their sexual organs and structures and sex cells. So what he did was able to remove the male parts from the purple plant. So in other words, that's the stamens, and he left the female part called the carpel left on the purple plant. Next, he went to a white flowered plant and took the male stamens with the pollen and brushed it onto the stamen of the purple plant. So again, what we're trying to do is introduce sex cells from one white plant of a pea plant to the purple plant to see what type of possible combinations we would get. And what you'll notice is that the pea pod down there in the middle, number 3, this is a mature pea pod and has many, many, oftentimes pods, or pod is the seeds. And so in this particular example, we have 1, 2, 3, 4, 5 seeds. And so each one of those seeds has the potential of maturing into a new plant. And so, as we look at what Mendel did, what he noticed is that 100% of the offspring from the purple and the white, the parent generation, were purple. And that was a keen observation. Again, there wasn't any exceptions. So we would call this purple plant, the parent, um, a purebred. And the same with the white parent, we would call that a purebred as well. So we had two purebreds and crossed them together, and we got all 100% um, in the first generation purple plants. So we call the first generation the F1 generation. You can notice that there. So then he did something unique. He took two of these plants and from the same generation, and he continued the experiment. So, what do you think if you hypothesize what would the second generation or the F2 generation look like? So, notice how we're using words like P for parent, F for the first generation, F2 for the second generation, and that's how we'll do it in the future here too. But what do you think? What would the plants look like if we took these two purple plants and crossed them together? And again, I would say that they're related. They came from the same parents. So in plants, it's a little easier to crossbreed ones that have similar parents or the same parents. And what you noticed here is that we got 75% purple and 25% white plants. So again, remember we went from pure white and pure purple to 100% purple. Now in the second generation, we get a mixture of 75-25. And again, this was done over and over again. So this wasn't just a one-time scenario. So again, Mendel noted this after hundreds of plants, and he got the same results every time, approximately 75-25 every time he conducted this observation and experiment. So why was that? Good keen observation on his behalf, and that's what we're going to discover. Why? So before we do that and look at some problems that you can do on your own, let's look at some terminology. In other words, some basic terms or vocabulary. And to understand the results that we just saw, we need to review some vocabulary. First of all, a gene is a chromosome or part of the DNA that codes for a trait or a protein. Now, that gene, we can look at our 46 chromosomes. We have around maybe about 20 to 25,000 genes. It's really unknown, but it's probably somewhere in this neighborhood of the 46 chromosomes. So you can imagine each of these chromosomes like a textbook. And this pair of chromosomes here has certain genes embedded in their DNA. So for each trait or gene, we'll receive one gene from the maternal side and one from the paternal side, from mom and dad. Now, the first term that I want to introduce is a term called genotype. And it's really a description of the gene, what we use using letters, like big H, little h. And when we bring this term genotype in, the other term that we use to describe some of the letters that we use and their form is something called dominant and recessive. And a dominant gene always shows up in the phenotype. In other words, it always shown up as the physical appearance. It's oftentimes referred to as the capital letter. The recessive is a gene that is hidden or can be hidden by a dominant gene. And it shows up only when the dominant gene is not available. And we oftentimes put this in a lowercase. So the example I have down below, I'm just using R's. So for example, we have red flowers and white flowers. What we'll typically do is the dominant gene will be risen as the uppercase letter, and I just chose red, so we'll use R's. That means that a white flower would use the recessive condition and we'll use little r. So the dominant red is going to be the capital, and the white is going to be flowered, is going to be the lowercase or r. These are referred to as our genotypes, and it gives us kind of an abbreviation as far as what it is that we're looking at as far as the gene. So one of the questions comes up is what is the dominant color trait for these flowers? Well, as I stated, the red was the dominant one, and it's typically the more numerous. Doesn't have to be, but it's typically the more numerous. And then why does the condition where you have a big R and a little r flower look the same color as the big R flower? In other words, if you look at this, you'll notice that each flower has two of the letters. So that means that each gene has two letters We refer to each letter as an allele. So alleles come in groups of two. So, for example, we have three possibilities. We have big R on the far right, which means that they have both dominant alleles, one from mom and one from dad. On the other side, we have a big R, little r, which is actually a hybrid or combination. One of the parents had a big R and one of the parents had a little r. So the offspring has a combination of both. And the third possibility is that the offspring, in this case, the flower, received both recessive conditions, the little r's from both parents, both mom or the mother and the father. So the combination here, if you have a big R, little r, as what dominance suggests, is that it will always be red, or the dominant condition. The dominant one will present itself, and the recessive, in that case, with a little r, will be hidden. So if we look at some of the genetic terminology and continuation, a gene is this DNA segment for one trait. And we could look at eye color, we could look at flower color, but it's some type of segment. So we call this, given a brief term, as an allele. It's a form of a gene. As I said, you could have brown eyes or blue eyes. Um, this gives us the genetic genotype that we're referring to, the genetic makeup of the DNA. And as I said for the previous slide, we have three conditions, which is noted down. So this is actually a code for what it physically looks like. And we give this term the phenotype. The phenotype is its physical appearance, whereas the genotype is the letters, the genetic code. And the physical characteristics could be a tall or a short or a thick hair, as was described here. So genotypes can come in a couple different forms. If they're both the same, in other words, both alleles from each parent are the same, we give this a term homozygous. And homozygous, the examples I could give you would be like a big B, or a little b. Those are two examples using B's and whatever B stood for. But we do know that they're both the same. And so both parents had the same alleles. And either you're big B, or another possibility would be a little b. That's homozygous. We oftentimes give this term as a purebred. In other words, like a golden retriever is oftentimes referred to as a purebred. Many of our animals that we have in our homes are oftentimes purebreds. The other term is referred to as heterozygous, and heterozygous is where you don't have both the same, you have some different alternate forms. So the alleles in this case are like big B, little b, a combination of both. So the big B came from one parent, the little b came from the other parent. Come together here, we have a combination of both the dominant and the recessive allele, and therefore the dominant will express itself. And if that was B for blue, that would be the one that would be expressed. And we oftentimes refer to this general term as a hybrid. So a hybrid is a combination of both for that particular trait. So phenotypes, just once again, physical description of the traits like Mendel observed, purple and white. And if we're looking at the genotypes and phenotypes for these flowers, well, then the phenotypes are going to be the colors, the purples and the whites. And the genotypes, well, we have to know which one is dominant. In our earlier discussions, it was shown that purple was dominant. So if you have a big P is the letters I chose, that would be a dominant homozygous. The opposite of that would be a homozygous white. In other words, we would abbreviate the genotypes as little p. And again, that would be the white would be the phenotypic expression of that. So homozygous have two of the same, and then heterozygous, two of the different. And that's how we're going to view this. The prefix homo means the same, and hetero means different. So now back to Mendel's purple and white flowers. If we look at the first generation, in other words, this is referred to as the P1, he crossed this purple flower with white flowers, and he got 100% of the offspring that were purple. And the original question was, well, why? Well, what we know is that the purple is dominant, and so those purple flowers, their genotypes, were big P, and he crossed that with the white ones, which were little p. And so every offspring received one big P and one little p. And therefore, we would refer to this individual as a heterozygous, and that the genotype would be the one that's being expressed, which is going to be purple. So his results, 75% purple and 25% white, dictated how that came to be. So this generation was entirely heterozygous. In fact, there was no other possibility. And the offspring, in this case, if we look at the F2 or second generation, could receive a combination of all three of those. And so you can have these kind of hidden within a generation, and then all of a sudden pop up again, sometimes, like, for example, in the F2 generation. So as we look at some of these Punnett, what are referred to as problems, the term that we use is Punnett squares. And a Punnett square is oftentimes used to look at the genotype possibilities. It's four squares. It's divided into four equal parts. We use letters to demonstrate the dominant trait and the recessive traits. So if I'm going to use freckles, for example, I'm going to say that freckles are a dominant trait, and non-freckles would be lowercase f, which would be no freckles. And what we would do is we would place the parents. For example, I'm going to have one parent as big F, little f. In other words, this individual is heterozygous. And then this individual will put on the other side or on the top here, and they are also heterozygous. And so what we're going to look at is some of the possibilities. Well, we just fill in the alleles. Remember, each one of these is referred to as allele. So we're going to take the big F, we'll put it here in the box and also down below. This big F would also go in this box and also this box here. We oftentimes put the dominant first and the recessive second. So in this case, the big F is going to be first in all four of those boxes, and the little f's are going to be second. Now notice here that it kind of came off to the side, but notice this one is little f. So in other words, this is a combination of one parent and another parent, and this individual would have no freckles. So this would be freckles, 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 and no freckles. And so what we're looking at is the whole square, 100%. In this case, one part is going to be 25%, and then we're going to start looking at some of the possibilities from that. As far as what are the ratios that exist here. So in Mendel's experiment, we look at like one trait. He observed many traits over the years, whether it's height or color. And each parent that he did, he made sure that they were purebred. So in other words, he had to breed them over and over again over time to make sure that they were purebreds for a particular trait that he was looking at. So for example, tall plants, he would breed them, keep on breeding them after many generations to ensure that all the individuals from those peas were tall. Same thing with short. So we call the original purebred parents the P generation. And on the left-hand side, tall genotype and short genotype, we can say, for example, a tall would be big T, and let's just assume that the short is going to be little t. Again, place him on our Punnett square and then filling it in. What you'll notice is that all four of the first generations, there's only one possibility, and that is big T. So the results of this give us the genotype and then the phenotype. The phenotype, in this case, it was kind of got messed up here a little bit. My apologies. But as far as the phenotype, they're all 100%, or 4 out of 4 are going to be showing that dominant trait. So what we refer to as a ratio, the genotypic ratio, is the number of big T's, and that's 4, and the number of big T, little t's, big T would be 0, but little t would also be 0. And this idea of the combination big T, little t, which is the heterozygous state, is going to be all 4 of them. So this created the first filial generation. And again, you have to excuse my numbers that got scrunched there. So Mendel wondered, does the short disappear for good? And as we said, it probably doesn't, just stays hidden. And so we took the F2 generation, crossed them together. We call this second generation the filial generation, or F2 generation. And again, he did the same thing, looked at the results of the genotypic ratio and a phenotypic ratio. And what you'll notice here is the one indicates the dominant condition where you had big T, big T. And then the two represents the genotypic ratio is big T, little t, which we had two of those. And then the one would be the little t. And again, I apologize if I have some numbers or some material kind of get crunched in here. So that's my genotypic ratio. But we would say that there's three of those, one, two, and three, are dominant. In other words, they're all tall. And there is one possibility, or one out of four, or 25%, that would be short. So it's a 3-to-1 phenotypic ratio. So these are Mendel's laws of genetics, as stated in 12.1. And what it is this he states a couple laws. The first one is the law of dominance. One allele can be dominant or the recessive. Number two, the law of segregation. Also found in 10.2, but the law of segregation states the following. In cells that have, or in organisms that have sex, they go through a process called meiosis, and pairs of these genes, or homologous pairs, separate. So, for example, if this is my individual from a parent, notice here that if this was the testes, for example, in human, or eggs in the ovaries of a female, we have a pair of these alleles, but they're going to be separated. And so if this has a total of 46 chromosomes, 23 are going to be in one cell, and the other 23 are going to be in another cell. And so these are going to be split up. So the big A in this case is going to be split into one cell, and the little a is going to be split into the other cell. Again, it depends on the combination. You could have homozygous dominant or homozygous recessive, but in this case, we just put a heterozygous state in there. But they are going to separate. And the other thing that Mendel noted is this law of independent assortment. In other words, during sex, species, these homologous pairs are going to separate randomly. They're not going to be distinct, whether it's from the male or the female. They are going to go into the sex cells randomly. In other words, you can think of it as this. If the green in this case are from the female and the blue are from the male, you can have all the blue on one side, all the green on the other, or an alternate is just mixing them up. And that's what this is really saying, is that there's going to be a combination and just a separation based on the randomness of these paired chromosomes. And that's the law, what Mendel says, the laws of independent assortment. And try these as an optional practice, and check with me if you want to see how you did with the purple people eaters and these practice problems on Mendelian genetics.